Okay, so we will get started with our second talk of the day, and actually our fifth one, so we're halfway done, which is all very, very exciting. And it's pretty much the same audience, so I won't remind you about the picnic or going onto YouTube, because you already know it's there. Anyhow, without further ado, we'll have the introduction of our speaker.
All right, so now it's my pleasure and opportunity to introduce Spencer Swick, another senior chemistry major who's graduating this year. Actually, biochemistry major. Biochemistry major that's graduating this year from Kimberly High. Go Papermakers. Spencer has quite a lot of experience with pain, working on our football team for many years and being injured in various and sundry ways. And so we talked a lot last term about how pain medication works and how we can make it work better. And his work and reading on that has been really impressive, I think. And so he's going to talk about what he's learned about different strategies for going sort of past the morphine paradigm in dealing with pain, both acute and chronic. So.
Hi everybody, thank you for coming. Like Professor Dever said, my name is Spencer Swick. I'm from Appleton, Wisconsin. And also, like he said, I've had a lot of injuries in my time. Just in the last 4 years alone, I've had 4 orthopedic surgeries and I've fractured my spine. So I've been in and out of the hospital every winter break since my senior year of high school, actually. So my talk, or the title of my talk, is Functional Selectivity, Understanding the Importance of Conformational Changes on Pharmacological Effects in Pain Management. So first, I'm going to start off with what is pain. So pain is a subjective experience that you feel when you have a harmful experience or stimulus. So there are 3 main types. First type is acute pain. That's when you're running through hollow and you stub your toe or something. Second type is chronic pain. That's stemming from an old injury. So in my case, I have cartilage damage in my knees. And so just because of that, there's a lot of inflammation there causing pain. And the last type of pain is called neuropathic. Neuropathic pain stems from nerve damage or a spinal cord injury, where there's being nerve signals sent without like an actual stimulus or a reason why. It's just kind of there and you're perceiving that pain. So how is pain perceived? So you have receptors that react to harmful stimuli, and those are at the end of nerve endings. They're free bodies, they're outside of the cell. And they respond to chemical stimuli. So if you go into Buffalo Wild Wings, you have the blazing challenge. That's a chemical burn essentially right there. Then you have mechanical pain, so that's you're stubbing your toe, you're tearing a ligament. And then you have your thermal pain, so you're touching a hot plate, not being very smart about that, so you have pain that way. And what these stimuli do is they create an action potential in the nerve center, or in the nerve body, which goes up A delta fibers and C fibers through the dorsal horn of the spinal cord. From there, it goes up the spinal thalamic tract to the nucleus raphe-magnus, and finally it goes up to the periaqueductal gray, or the PAG. And the PAG is actually where we have most of our drugs are being focused on for treatment. There you can stop the pain both going down the spinal cord as well as stopping the nerves from firing up to the brain, where the pain is perceived by the amygdala, the hypothalamus, as well as the thalamus. So, a little bit about nerve transduction. So we have an axon, and you have action potential where sodium channels open in the cell, and sodium flows into the cell. And then when that happens, we also have potassium channels open, and the potassium diffuses out of the cell. However, the potassium diffuses out of the cell slower than the calcium is flowing in. So we have an internal cation increase, and that's what you see with these charges slipping here. When there's a difference in charge of greater than 15 millivolts, the nerve fires, and you have an all or nothing, so it either fires or it doesn't. And from the action potential travels down to cell to the synaptic cleft, or the nerve synapse. So this is where all the magic happens. So we have an action potential reaches the axon terminal, and then that triggers calcium channels to open, and this calcium ions flow into the cell, where they bind with vesicle proteins, and the vesicles will then go to the synaptic cleft, and they will be released into the synapse. And then these neurotransmitters will cross the synapse into neuroreceptors on the other side, so it's the next nerve in line. And from there, the trigger is sent further on. So how do we manage pain? First off, you can prevent the action potential. So that's if you're blocking your sodium channels and your potassium channels here. And the other way you can do that is actually blocking the calcium, or the signal transduction, by preventing calcium from flowing into the cell so that you can't have neurotransmitters being released, so you're stopping the signal right there. So the analgesic pathway, we have a 7-transmembrane G protein coupled receptor, which is the opioid receptor. The opioid receptors actually have 3 different subunits. You have a kappa opioid subunit, you have a delta opioid subunit, and you have a mu opioid subunit. And so when a ligand binds to the receptor, it activates a G protein phosphorylation, which causes a decrease in intracellular calcium. So those calcium ions are not flowing into the cell. And this causes a decreased neurotransmitter release and ultimately inhibition of nerve signaling. So now I'm going to transition to a little bit of the history of pain management now that we know how it actually occurs. So the first one is opium. It's cultivated all across the world. It's been known by the ancient Sumerians as far back as 4000 BC. However, it was first accurately described in about 300 BC by a Greek philosopher known as Theophrastus. And this latex here, which they take from an unripened seed pod where they just cut and then the latex falls out, is actually the opium that they harvest. In there, we have over 20 different alkaloids, which are just nitrogen-containing compounds. And then these were first isolated in the beginning of the 19th century. So first is morphine. This was isolated in 1804, and it's actually the most potent natural analgesic that we know. And then scientists from there have taken morphine and they've made derivatives, they've adjusted it, changed it into things such as oxycodone and hydrocodone, which is your Vicodin, that's your Percocet. And if you're just looking at the structure, you can see how they're very close. We have this section right here is known as the pharmacological. The only difference is, obviously, we have a hydroxyl group instead of a hydrogen there. And we have a carbonyl group instead of hydroxyl group, and then the methoxyl group instead of another hydroxyl group there. And so these different changes, so the pain management drugs today, we have obviously advantages. You have the pain relief, the analgesic effect, but we also have a lot of different disadvantages with current pain drugs. For instance, there's a very easy to build up tolerance, and there's also withdrawal symptoms. There's mood changes, there's sedative effects, and there's also you lose motor control, so you're feeling really slow. It's the reason why you don't drink and drive, essentially. It's very similar to that. And so, because we have a receptor, it needs to be regulated naturally. So what happens is that we have a protein called beta-arrestin. It's a small protein family that regulates GPCRs, both for opioid receptors and other receptors. It causes a desensitization of GPCR through a process called internalization. So when we have a ligand binding to the receptor, we have a phosphorylation event, which ends up recruiting beta-arrestin to the receptor. And then this beta-arrestin ends up recruiting AP2 proteins and clathrin-induced pits. You see here, and you can see that the receptor is starting to be pulled into the cell. So this happens, and then we have dynamin-dependent fission, where this cell, or the receptor is actually being endocytosed into the cell. So now that the receptor is surrounded by a vesicle in the cytoplasm, instead of on the plasma membrane where it can continue to be activated. So beta-arrestin, besides causing a desensitization of the GPCR, will also start signaling cascades for different pathways. And that's ultimately where we have a lot of our negative side effects come from. So far, I've talked about how pain is transmitted and perceived by our bodies. I've also kind of went over some of the unintended consequences of our current pain management drugs. And so what are we doing to combat these drugs is what I'm going to start talking about next. So we have something called a G protein bias agonist. It initiates a certain conformation in the receptor, which produces a more specific response, which makes it easier for drug development and drug control, so you know exactly what's going to happen. And if you look at this picture here, we have a G protein bias agonist come in, and if you look at this section of the receptor, you notice that it's kind of tilted at an angle, and that's the pathway that leads to analgesia. But when you have an unbiased ligand come in, you see there's not that not that tilt there anymore, and the recruit's beta-arrestin. You see how just those small conformational changes can have different effects on the cell and the signaling pathways. And so this is kind of an example of these conformational changes. We have nolorphine, which is a morphine derivative on the left, and we have morphine on the right. So nolorphine, instead of this methyl group, has a butene group, and this actually changes the structure of the receptor around it. And if you look, you can see kind of how this shape is different from this one. And I don't know how well you can actually see this, but actually if you look at these blobs, these are amino acid residues. They're specific amino acid residues. And so we have tyrosine 139 right here, and we also have tyrosine 139 down here. So you can see very easily how the receptor is a different shape based on the type of ligand that's bound to it. So now current drug development. First drug is RB64. I found this drug from a paper from the Roth Research Lab at University of North Carolina, Chapel Hill, Department of Pharmacology. It's derived from salvinorin A, which has been shown to be a kappa opioid receptor agonist, an unbiased agonist, so it will recruit beta-arrestin. So it's shown to be a kappa opioid receptor, which is one of the specific receptors. It controls calcium channels, and it does not recruit beta-arrestin. Then we also have INTA or INTA, which is a loose morphine derivative. It's actually KOR-DOR agonist, meaning that there's a heteromer, those two receptors are actually working together, and this drug is acting on both of them essentially at the same time. And this also controls the calcium channels, as well as it does not recruit beta-arrestin. So first off, I'm just going to talk about both these drugs in terms of their analgesic effect as well as their tolerance. So to test the analgesic, RB64 use a hot plate assay where they stick a mouse onto a hot plate. It's a maximum of 30 seconds, so there's no permanent damage to the mouse. However, they did, when the mouse starts showing signs of distress, where they're jumping up and down, or actually if they urinate on the hot plate, they take them out, and that's how much pain they can tolerate. And for the INTA assay, or the INTA drug, they use the tail flick assay. Essentially the same thing. They're using heat from a light source. They're generally focusing a light source onto the mouse's tail. Same thing, if they flick their tail, they move out of the way. That's considered a response to the pain, and that's when they stop timing. So RB64, they had a baseline of about 15 seconds, and it does show an analgesic effect. In this paper, they actually they compared RB64 to two other drugs, UZX Iac5o5o5 and salvinorin A, which are both opioid receptor agonists. However, they're not biased, so they are recruiting beta-arrestin. They're more in line with morphine. So if we look at U69 here, you see that there's a large increase in the analgesic effect and the pain tolerance threshold. When we look at RB64, you see the same thing, going up to about 150% of where they are, or 50% greater than where they already were. And they found a subcutaneous effective dose of 3 milligrams per kilogram. So when you look at INTA, they used the tail flick assay, and they found that there was an analgesic effect. They found an intrathecal effective dose of 21.07 picomoles per mouse, and they actually went on to find a subcutaneous effective dose of 0.97 milligrams per kilogram, which is actually 9 times more effective than what morphine is for a subcutaneous injection. And then we also found an oral effective dose of 9.08 milligrams per kilogram, which is actually, unfortunately, 4 times less potent than morphine. So we do have this analgesic effect, which in one way it is better than morphine, and in other ways actually worse. But we also had no tolerance buildup, which is very important. This black line on the graph corresponds to the first day that they tested the pain thresholds to see which dose was effective. And then they tested 3 days later, and this is that red line, and they found essentially the same dose amounts were still effective 3 days later, just kind of showing that there wasn't any tolerance buildup. So next, I'm going to talk about withdrawal and mood changes. So for both drugs, they use the conditioned place aversion. This testing for withdrawal symptoms based on an unconditioned negative response to being placed in this box. So when a mouse is placed into this box, they're first originally tested in 3 phases. So the habituation phase, they're just letting the mouse run around. There's no divider. They're waiting to see where the mouse spends most of their time. If they're spending too much time on one side or the other, the mouse is removed from the study because it's showing a bias before they're actually having a stimulus. And the next stage is conditioning. They did this over a course of 10 days where every other day they would inject the mouse with the drug, and then the next day they would inject it with a saline solution, and then they would place them 30 minutes later into one of these boxes to see where they were placed into a box, and only on one side they weren't allowed to roam around, so they were being conditioned to have an association with that side of the box. And then finally they did a test phase where they just put the mice in the box, let them roam around, and they measured to see how much time they spent on each side of the box. If they were on the side where they were being conditioned with the result or with the test, or if they were on the other side avoiding the box that they were being placed in when they were being tested. So RB64 was the first one. They did actually unfortunately find a version, and this is the control, just the saline solution mice. You can see there's very little aversion as you go down. So just kind of explain this. Further you go down, is the further amount, or the more amount of time that you spent away from the side of the box that they ended up associating with a negative experience or withdrawal symptoms. And then these last 4 lines, these 2 are 1 milligram per kilogram dose of RB64, and they found that there wasn't any statistical significance with that based from the control, so they didn't have any aversion that time. But when they used the effective 3 milligram per kilogram dose at the end, they did find that there was a negative association with that box, indicating that there were withdrawal symptoms. And then INTA, same thing, except in this case, if they were above the line on the top, that they spent more of that time inside the box that they were being conditioned to necessarily avoid. And so there aren't any withdrawal symptoms because the mice spent actually more time on the side of the box that they're being conditioned to essentially avoid. And this actually is kind of pointing towards a reward pathway. They're associating something positive with that side of the box, so they're spending more time over there. And that's kind of what I was talking about with like the mood changes, where they're almost having a euphoric effect. I talked to one of the researchers for this paper, and they actually found that this drug actually also works pretty well as an antidepressant. So that's kind of a cool little bird with that as well. Yeah. So next I'm going to go into a depression assay for the RB64. They're using an intracranial cell stimulation where they're hooking up an electrode to the mouse that's connected to the pleasure centers of their brain. And these mice are then conditioned to hit a button here, which sends an electrical pulse up through the wires into the brain to the pleasure center. So if you shock it, you feel really good. It's awesome. And so from there, they didn't actually show any depressive effects. With the baseline, just a saline solution here, you can see as the frequency of the electrical pulse increased, the response rate where they're hitting the button more to get more of those shocks for pleasure increase. And here we have RB64, which is slightly less than the saline solution, but it is still much higher than both Salvinorm A and the UZX Iac5o5o5o. And then next, I'm going to talk about sedation and core motor control, so those other effects. So the RB64, they place the mice in this box to measure total movement, which is called a novelty-induced locomotion. And they're given a drug 15 minutes prior to being placed in this box. And this box has light sensors that measures how much the mouse is moving around in that box over time. And in this case, they left them in there for 30 minutes, excuse me. With our control saline mice, they found that they traveled just over 4,000 centimeters in 30 minutes, where the other unbiased agonists produce much lower effect, showing that there is a sedative effect involved with those drugs. And then finally, we have the RB64 at the end, which is showing roughly the same amount of movement as the control mice, showing that there isn't any of those sedative effects for this drug. And then next is the poor motor control. So they use a rotor rod assay, which you can see a picture of here, where they place the mice just on a rotating spindle. I kind of compared it to the lumberjack games with the guys that are running on the logs. And it's just testing your your coordination motor control with a baseline of about 300 seconds for these mice. They found that it does not impair their coordination. For a 3 milligram per kilogram dose, they found that the wild type mice, which are mice that don't have any genetic modifications, and then there's the beta arrestin 2 knockout mice, which have been genetically modified so they aren't expressing the beta arrestin, so they know that the beta arrestin is not what is responsible for any effects that they see. And in this case, between the mild type and the beta arrestin 2, you can see that there wasn't any difference the amount of time that they spent on the rod compared to the baseline. And even when we increased the dose from the effective 3 milligrams per kilogram to 10 milligrams per kilogram, there still was very little difference in the amount of time that they were spending on the rod. So that's just indicating that there wasn't any of those poor motor control side effects. So RB64 is analgesic. There wasn't any tolerance that was actually shown. I forgot to mention that, but it wasn't. They didn't actually have a graph in their paper, and just kind of said it. But they also didn't have any depression symptoms, and they also had no sedation, and no motor control deficits. And then INTA, we had an analgesic effect as well. There was no tolerance buildup. There was no withdrawals, and we might have even had an euphoric response instead of a depressive response. And unfortunately, RB64, they did have withdrawal symptoms, so if they continue forward with this, that's something they would have to fix, so they'd end up using a different drug more than likely. And then INTA, they did not test sedation or motor control in this paper, and I haven't found anything else on this drug. This paper is actually brand new. Or not brand new, but it's from November 2014, so just past November. And RB64 is actually just from February the paper was published. So future directions for both of these drugs. RB64 is actually not being tested further. It was actually used more of a proof of concept, and the research group is looking into similar compounds for improved benefits. And then INTA, it's being further tested. They have submitted a patent application for both this drug and several others that they were working on in this paper as well. This is just the most promising one. And unfortunately, there's a lack of funding currently, so they're not going very far at the moment. However, they're obviously trying to work on that as time goes on. So our conclusion, so receptor confirmation is a very important aspect of drug development. Being able to know how a drug is affecting the conformation of the receptor can lead us to understanding the signaling pathways that are important to drug development, so we can develop drugs that are affecting only a specific pathway, not activating multiple ones, like you see with morphine, where you're both getting the necessary and wanted analgesic effect, you're also getting a slew of unwanted side effects. So if we can find something that causes a specific conformation in a receptor that causes that only starts a single signaling cascade, we'll have drugs that will work much more effectively, with much less side effects. So thanks for attending, and do you guys have any questions? Yeah.
Would it be possible to maybe modify, say, something like morphine into something that's more selective?
That's essentially what they did with the INTA, and there's some other drugs, such as oxycodone, hydrocodone, where they're directly modifying the morphine into these other derivatives. However, they haven't found one directly yet. The most common ones still have the beta arrestin recruitment that's causing these other side effects. However, that's something that they're always kind of doing. They're slowly adjusting different morphine molecules, or going off of different drugs that they've already made, such as like RB64 or INTA. They're just slowly adding changes to see what works, what doesn't, what improves like the analgesic effect, or what decreases it, those sort of things. Anybody else?
I think you kind of addressed my question in saying that the paper just said about the tolerance, but I was very curious about this testing for 3 days. Is that a standard, like if the mice don't develop withdrawal symptoms within 3 days? Would they develop symptoms if they were on morphine for 3 days without withdrawal?
The way they made it sound in the paper is that what you would have those withdrawal symptoms. My uncle actually is on morphine, so besides just my own personal experience, I've had a lot of family that are consistently in pain, more so than myself. So that's also kind of driving this a little bit. But the way he kind of has made it sound is like you can definitely tell when he hasn't taken morphine. Like you can just see he gets really grumpy, irritable. Like you just kind of don't want to be around him at that point. And that's only after a couple of days. Granted, this is also after he's been using it much longer.
That's what I was curious about.
I don't know if the 3 days is for sure. I didn't actually look at that, but I would assume so.
I mean, I'm just curious if you have to be conditioned to it first, and then if you take it off, you don't see it work at 3 days.
Oh, with these, with both of those assays, they're actually using drug-naive mice.
Okay.
Yeah, sorry, I didn't say that when I was doing that, but yeah, all these mice have never been injected with any of these drugs, so this is all brand new to them, essentially.
And you think that if you did the drug-naive mice with morphine, that within 3 days you would see those symptoms?
Yeah.
Okay, we're going to ask about the mice. For little things. Can you go back to, there was one slide where you were looking for mice on a top plate?
Yeah.
Anything but the data was interesting. Little thing. There was actually, keep going back. It may have been with their poor little tails.
Okay.
Nope, there it was.
This one?
Yes.
Okay.
So after 20 minutes, it seems like, what is this WT vehicle that it seems that like they decrease the percent. I'm looking at the lowest pink one on the 20 minutes.
Here?
Yeah. So are all the other trends are going up in those two?
So both of these, they're just injecting, it's just like the solution, the drug's not actually in there.
Oh, okay.
Yeah, it's just what the vehicle that they're putting the drug in, the solution that they have the RB64 or these other drugs dissolved in. So in that case, it's only the vehicle that's just kind of showing that there's a baseline, that there's not.
Gotcha.
Yeah.
So 100% is just bad.
Yep.
Okay.
Yeah.
Gotcha.
Yeah. Anybody else?
What are the N values for these sorts of things?
I'm sorry, what was that?
The N values, how many mice do they test these on?
Please don't say it if it's a lot.
I think for both of the assays, for the analgesic assay, it was just over 10. It wasn't like a whole lot, but yeah, I didn't look too hard into that actually. Don't remember. Yeah.
So, so ideally we'd like to find more compounds that sort of bias the G protein where you want.
Yeah.
And the two drugs you looked at, one is a pretty clear morphine drug, the other's not.
Yeah.
So is this amenable to some in vitro screening assay where we can look at a lot of things very quickly?
Yeah,
Do you get a sense of how these things,
Wait,
like
you said-
how in vitro tests would work here? Like before we get to humans, could we do this in some plants?
Yeah, they did that a little bit in previous papers. Both this research group, they were one of the graduate students that was working with this. Also had published a paper a couple months prior to this one, where they're also going through RB64 and different scaffolds for the receptors, and then there was also. I forget what was going on. Can you repeat your question?
But this does seem to be amenable to some screening.
Yeah,
process.
they actually ended up screening all these drugs prior to actually testing them with the mice through in vitro testing, just to see that I know specifically for RB64, they tested to make sure that it was only activating the G proteins in vivo, in the testing. Yeah. Just to make sure that it actually was only activating the G proteins and not recruiting any beta arrestin.